So now it is my distinct honor to introduce Dr. Barry Kreiswirth, a distinguished member of the Center for Discovery and Innovation at Hackensack Meridian Health. Dr. Kreiswirth's academic contributions extend to his roles as an adjunct faculty member at the Department of Medicine at New York University, a lecturer at Columbia University's Mailman School of Public Health, and a visiting professor at the Museum of Natural History. Dr. Kreiswirth earned his PhD in microbiology, focusing on the genetic basis of Staphylococcus aureus toxic shock syndrome toxin. With a career spanning over 4 decades at the Public Health Research Institute, he has pioneered molecular epidemiology, particularly in understanding the spread of antibiotic-resistant bacterial pathogens in hospital settings, as well as in the community. His laboratory developed rapid diagnostic tools and implemented SPA typing, which is sequencing of the polymorphic X region of the protein A gene, to genotype methicillin-resistant Staph aureus, methodologies that have since become gold standards in supporting hospital infection control efforts. In response to the multidrug-resistant tuberculosis outbreak in New York City, Dr. Kreiswirth established the Tuberculosis Center, collaboratively working with the CDC and health departments to control the spread of tuberculosis. The center notably characterized the highly multidrug-resistant W strain and created the largest Mycobacterium tuberculosis strain and DNA fingerprint library in the US. At the Center for Discovery and Innovation, Dr. Kreiswirth's laboratory focuses on controlling infections in high-risk cancer patients and transplant patients, bringing molecular diagnostics and epidemiological studies to advance infection control efforts across the Hackensack Meridian Health Hospital Network. With over 435 peer-reviewed publications, his expertise is widely recognized, and he has been featured in National Geographic and on 60 Minutes. Among his many honors, he did receive the Waksman Award from the Theobald Smith Society in 2015. So please join me in welcoming back Dr. Barry Kreiswirth.
Okay, thank you for the invitation. It's very nice to be here. It's early this morning, I know. Hopefully everyone has had coffee, including me. So what I'm going to talk to you this morning about is a study we've been doing for the last 1.5 years on looking at drug-resistant Klebsiella pneumonia. And as you'll hear, this is really a study that focused on the community, which is a little bit unusual for most epidemiological studies. So I'm going to touch upon, obviously, this is a long list for a short talk, but I really want to give you a little snapshot of each of these topics. So clearly, the big theme is the molecular epi of drug-resistant Klebsiella pneumonia. The focus of this study is really, or the importance of this study is really our collaboration with Quest Diagnostics, as you'll see as we go through, who does capture the community and a lot of outpatient populations. Excuse me, I do have an allergy. As you'll hear, this study is mostly about UTIs in elderly women. We have a very, very large plasmid epidemic that I'll try to give you a sense of how this is happening and our understanding, which is still limited. Ultimately, the take-home message today is that we do have a major problem in drug-resistant Klebs that are difficult to treat, not only in the hospital, but in the community. And ultimately, this epidemic is caused by the spread of one gene, CTXM15, which is spreading both as a plasmid epidemic as well as a strain spread. And ultimately, hopefully you'll appreciate that our lack of surveillance, which is getting worse over the years, is really the key in understanding the extent of this problem. Okay, so I think it's easier if I talk this way. So obviously, as you all know, antibiotic resistance is a major problem. It's always been a major problem from the beginning. These numbers are just constantly being sort of thrown around. But at the end of the day, as you know, the biggest problem is we have drug-resistant organisms and the drug companies are not making new antibiotics. Okay, so what we're going to focus on today is really the focus on beta-lactams. So beta-lactams, this is an old slide, but the numbers have not changed over the last 10 years. we still treat most patients with the beta-lactam. And that history goes way back to penicillin and to the discovery by Fleming and the licensing of penicillin back in 1945. So this problem continues to arise. As you'll see, drug companies have made new beta-lactams and the organisms have developed resistance. And this is this constant battle that goes on for the last 80 years. Okay, so this is really, not sure. Okay, anyway, this goes back, as I said, this is an 80-year history. And as you'll hopefully recognize, the evolution of drug resistance bacteria has been a really small window. 80 years over time, we've gone from the discovery or the licensing of penicillin all the way to the development of a novel drug called ceftriaxone. What I want you to appreciate is when you look at something called first-generation cephalosporins, this is an example. The drug companies over the years have made literally tens of different, each of these classes. So there are hundreds of different beta-lactamans that have been licensed over these 80 years. But the consistent problem is that as we get resistance to one class, the drug companies develop the next class or the next generation. And this has just been going on ever since 1945. Okay, so this is the battle. And what's remarkable, and literally every time I look at these numbers, there are people who actually do this. They keep on looking at the literature and just updating this. If you don't believe in evolution, just look at this number. In 80 years, bacteria have actually, mostly acquired 2,770 beta-lactamases. So you should appreciate that this is an ongoing problem. It's not going to go away. The bacteria are not going to give up. They're survivalists, and this is just how they do it. They constantly are ahead of the game, and unfortunately, we really have fallen behind. So I'm going to give you now some examples, just really to give you an impression of what the problem is and what our lack of understanding is until a lot of studies needed to be done, mostly based on surveillance. So this is the first level. We had this development of first, second, third generations. And when we developed ceftriaxone, which was developed by Roche, ultimately, ceftriaxone is still, to this day, a remarkable antibiotic. It's an injectable. It really is a powerful drug, and it's really the workhorse, and we still routinely use it. The problem was that in the mid-1990s, Klebsiella, E. coli, a lot of the enterobacteriales ultimately developed resistant to third-generation cephalosporins. And when they did, what happens is that everything before that is not a useful drug. So if you have what's called an ESBL, which is an extended spectrum beta-lactamase, you cannot treat with a third, second, first, or penicillin. So it's literally wiped out those classes of drugs. So that was a problem. And this, as you'll see, is the cycling. So we had resistance in the mid-1990s, and the drug companies then got on top of it. They started to develop fourth-generation cephalosporins, and cefepime became sort of the model. And then there was piptazo in 1993, and then the remarkable development of carbapenems. So when we ultimately, as the Klebsiella, ESBLs, and E. coli's became a problem, the drug companies were on top of it. And once carbapenems were developed, everyone thought this problem was over. But it's kind of naive, because all you have to do is look at the history. It's clear that even though carbapenems were really powerful agents, and we didn't seem like the bugs had become resistant, you really have to be a fool not to appreciate the fact that it's just a matter of time. And in fact, that's exactly what happened. So around 2005, we started to see slowly the emergence of resistance to carbapenems. And not surprising, the organism, this was mostly E. coli, mostly Klebsiella, although E. coli and other gram-negatives have acquired this. These are, again, beta-lactamases, carbapenemases, that were acquired, and ultimately now ESBLs and carbapenem resistance is now a problem. So again, we're now at the point where what do we do against carbapenem resistant Klebsiellas? And that was a real gap. And ultimately, from about 2010, when it really started to spread, and then really from about 2015, these were serious problems where literally we could not treat patients who had carbapenem-resistant Klebsiellas, and the mortality increased, and the drug companies were definitely behind. What was interesting about that epidemic, which has been very well studied by us and other people, was we realized it was really the emergence of one major clone. So as you may know, lots of drug-resistant bacteria like MRSA, E. coli 131, there are unique clones that do emerge. Well, in this case, ST258, which is not even a common Klebsiella, acquires a resistance plasmid that carries a carbapenemase, a KPC, and this one strain literally dominated the US medical system. It spread, it represents about 80% of all carbapenem-resistant strains in the US, but then it spread from the US, it went to Israel, it went from Israel to Italy, Italy to Greece, and literally this is a clonal spread that started mostly in the Northeast here in New York and New Jersey, and now has become the predominant clone. Okay. Now, this is the part of microbiology and drug resistance we never understand. We never understand why things slowly just disappear. And I say disappear, I don't mean that literally, but we did start seeing, almost coincidence with the COVID epidemic, all of a sudden carbapenem resistance seemed to have been under control. We were collecting 30, 40 isolates from New York hospitals per month, and all of a sudden, over time, we're seeing 1 or 2. Now, why? Well, it's always hard to figure out why something seems to not go away, but seems to be controlled. So you have to add up a number of factors. The one thing is that's definitely happened was our diagnostics got better. We could identify carbapenem-resistant Klebsiella a little bit faster, but most importantly, the drug companies figured out a clever way. So what the drug companies have started to do is they started to realize, and this is almost out of survival, that if we have so much beta-lactamase resistance and we can't use the beta-lactam, which has already been an effective drug, what can we do to sort of bring back that drug? And the idea was, well, maybe if we make inhibitors to the beta-lactamase that the bug has, then we could partner that beta-lactamase with the beta-lactam. So literally you're doing is you're counteracting the bug's ability to produce an enzyme, you're inhibiting that enzyme, which allows you then to utilize the drug again. So this idea of using inhibitors with a beta-lactam has become somewhat of the approach the drug companies are using. And that happened with what was called, it's cefazidine and avibactam. That combination really does work against carbapenem resistance. So that was one really major advantage now for the doctor's arsenals. And then, as I said before, a new drug called cefidrocol, which is a really interesting drug and a novel drug, really, I think, helped at least give the doctors an arsenal. Again, these are hospital pathogens. These are all injectables. But between diagnostics and these 2 new agents, we did seem to get on top of the carbapenem resistance. Now, not surprising, we do have resistance to Cas-Avi, which is the combination. Some of the KPC enzymes have actually changed and now are actually resistant, but that's rare. So the good news is the carbapenem epidemic seemed to be under control. But as this is going on, and it's like anything else, we sort of took our eye off the ball, because why carbapenem resistance seems to go down, ESBL, which again started in the 1990s, has not gone away. In fact, in the background of the carbapenem resistance epidemic, ESBL has just constantly rose. And if you look at any hospital studies, it's always been trickling up, and it's sort of been something we just don't appreciate. And again, the reason is, well, we can treat them, right? We have carbapenems now, we have other drugs, but we really don't understand the extent of the CSBL problem, which, as I said, keeps on just growing. Okay, so that's where we really were when we started to think about this problem. And we said, you know, we don't really know a lot about ESBL-resistant Klebsiellas. It's sort of jumped the radar screen when we got carbapenem resistance, and we never really looked at the molecular epi. So that was really what we started to think about. Well, maybe we need to do a study and understand how bad this problem is in the United States. And the problem with doing this study is, again, most of the data that we had historically was from various hospitals around the country. So we decided that could there be a better way to really capture a large population and just do a snapshot? And so what we appreciated was that clearly all the SBLs, as I told you before, are resistant to these first, second, third generations. So our idea was to basically see if we could identify a collection using whole genome sequencing to really just understand what is the extent of this problem, and is it a problem? Is it like ST258 where we have a clonal spread that we just don't appreciate? So to do this study, we had one luxury where our new campus is. So CDI is now in Nutley, New Jersey. It's on the old Hoffman Roche campus, and one thing that has been developed over the years is that Quest Diagnostic moved from Teterboro, and they have literally a brand new facility that sits on our campus. Quest Diagnostics, I'm sure you've seen these small little white trucks that go all around the state. They're a national diagnostic laboratory. So they have labs all around the US. They also have labs in Europe and Asia. But the main lab for the Northeast is in Clifton or Nutley. It's on the same campus. And literally they get isolates or primary specimens from Montreal all the way down to Virginia at that site, with the exception of Pennsylvania and Massachusetts. So it's a large collection. So as a neighbor, I started to visit them and started to talk to them and said, basically, you know, what you throw out every night, I want. And that was basically the idea of the conversation. They literally do 180,000 microbiological tests each year. So they have an enormous collection of organisms that, as I said, they literally throw out each night. Just as an example, they do 7,000 urines a night. So it's a huge reservoir. So in our conversation, I said, you know, we could do a very simple study. If you're willing, we could ask you to identify ESBL-resistant Klebsiellas, send them to us, we will sequence them and try to understand what is going on in the US. And that's exactly the study we did. We identified 10 laboratories, as you'll see in the next slide, from around the country. We asked them when they saw a Klebsiella-resistant strain that was ceftriaxone or cefazolin resistant, which was indicative of an ESBL resistance, they should capture it and send it to us. And the only thing we knew about the isolate was the site of infection. We knew the antibiogram, we knew it was a Klebsiella, and we knew it was third-generation resistance. And that was the nature of the study. And it went on for 1 year. One of the problems with molecular epi studies, and the CDC is notorious for doing this, is that if they do a 1-year study, by the time they publish it, it's 5 years after the fact. And that's really a problem because it's not very useful to have 5-year-old data for things like drug resistance. So one of the challenge was if we could do this study and collect everything in a year and get a large snapshot, our idea was to turn this around by sequencing and doing the analysis, hopefully within a year later. And that's what our goal was. Okay, this is a very complicated slide, but it really summarizes a lot of work that we've done over the last 1.5 years. So on the bottom, you can see where the stars are. So here's the one in New Jersey. Every star represents just a different diagnostic site of Quest. So they literally are scattered around the country. There's more Quest sites than this, but these are the large ones. And again, you can see the numbers. So one thing you should appreciate, during this 1 year, they actually cultured 20,000 ESBL-resistant Klebsiella. We captured about 2,040, so we have about a 10% capture. This is not epidemiologically comprehensive. It's a snapshot. The most interesting thing, which we didn't appreciate when we started the study, was how much they collect as outpatients. Now, as you know, because there's sites all over the state, you can go to a Quest clinical site and they do testing. They'll send the organisms or the primary specimens to Clifton. And ultimately, the capture is the outpatient population. So we're really looking at community isolates. 82% from the Quest database is what we saw in our collection. So this is not a hospital-based study. This is a community-based study, which was important. Now, in terms of who they are, 67% were from female, and the age average was about 66 years of age. So you have basically elderly women in this population, and when you put that together, not surprising, 90% of these Klebsiellas are urinary tract infections. So what we're seeing is a highly drug-resistant population, primarily cultured from elderly women throughout the United States with urinary tract infections. Now, in terms of the first question we wanted to know, which was, is this a single outbreak like ST258? The first answer was no. And in fact, if you can see this, ST307, which is another known Klebsiella genotype, represents about 20% of this collection. That's the largest group of isolates in this study. It was remarkably diverse. We ended up finding 120 different sequence types in this collection. So in contrast to the carbapenem-resistant outbreak, this is not clonal. This is a really diverse group of isolates that are all ESBL resistant coming from the community. So it was very different just from really looking at just our collection before we even started to analyze them. Okay, so this is a complex antibiogram for about 70% of these isolates. I don't expect you to look at and know, but each of the lines across is just a different group of sequence types. The point of this slide, the take-home message of this slide is clear. Now, also, so what we're looking at here, and this comes right from the clinical data from Quest isolates, you're looking at resistance, intermediate, and susceptibility. One thing you can see is not surprising. On top left, they're all, if not 100% cefazolin resistant, the majority are ceftriaxone and cefazolin resistant, as we expected for ESBLs. Cefepime, which is the fourth generation, is about 50% treatable. Piptazo is a little more susceptible to piptazo. And as expected, they're treatable with carbapenem. So both imipenem and meropenem, which are carbapenems, ESBL are susceptible, as I said before. The surprise and the concern is the bottom row. So the bottom row has three drugs: nitrofurantoin, Bactrim or trimethoprim sulfathiazoxazole, levofloxacin, and Cipro, which are quinolones. What we know about those three drugs, those are all the oral drugs we use to treat urinary tract infections. So if a woman wants a drug, calls the doctor, not even culturing it, the knee-jerk reaction is to give someone a fluoroquinolone. Well, that's a problem, because if you add up this resistance, about 75% of these isolates are resistant to all three of these oral drugs. The only drug that's an oral drug that's really useful in this setting is fosfomycin, and it turns out most of these strains have Fos-A in their chromosome, which gives you fosfomycin resistance. So the problem is, just from this data alone, is that most of these patients cannot be treated with an oral drug. They're going to have to see some type of clinic or go to the hospital and get an injectable. And that's a major problem. And this is not going to get better. This is only going to get worse, as you'll see. So this was our first surprise. This is really, out of all the findings in this study, this is the most clinically important and the most clinically scary, because this is only going to get worse, as you'll see in a second, because the resistance, as we now determine, is spreading. Okay. So this is the molecular data. So as I said, we sequenced 2,040 isolates by Illumina, and the goal was really to identify the sequence types that are predominant. And that's a tree up top. The take-home message, as I told you, was we do have a lot of diversity. ST307 represents the most common strain. So that's the one on the left under B. But when we started to look at mechanistically, what do all these strains have and why are they all ESBL resistant? The thing that really came out very quickly was they all seem to have CTXM15, which is the enzyme that they acquire, and you'll see what I mean by acquire in a second. It's the enzyme that literally almost 92% of these isolates all have. Now, what's interesting about CTXM15 is that's just one of many CTXMs. The history of these ESBLs since the 1990s is the evolution of different CTXM enzymes. And there's literally tens of different CTXMs. CTXM15, and we don't know why, has emerged. And it's now the dominant ESBL resistant determinant. And as you'll see next, it's also plasmid mediated, which is obviously a problem in terms of its spread. If you look at the bottom right here, this is just a heat map showing the fact that when you do Illumina sequencing, you cannot assemble and really determine all the plasmid genes. Illumina will tell you what genes are present in the strain and which plasmid replicons are present in the strain. So if you look on the right here, this is just really a cartoon to say how many plasmid replicons are present in our collection. And on average, Klebsiella ranges from having 2 plasmids to 5 plasmids, and the mean is somewhere between. But what we also realize, and that's the first column in the heat map under C, almost every strain has an INCF plasmid. And as you'll see next, these INCF plasmids are enormous plasmids. They're over 200 kb in size. And it seems like 90% of our isolates have an INCF plasmid. So, and as you'll see when I show you the cartoon of the plasmids, these are hybrid plasmids, which means they have 2 different replicons, which allows them to actually move better because they can get around plasmid incompatibility. So this really gave us an insight that we have a CTXM15 problem, and it seems like they're hitchhiking on these INCF plasmids and spreading to lots of different genetic backgrounds. Okay, so as I mentioned before, if you do Illumina sequencing, you can understand content, but you can't assemble the plasmids. It's almost like when you make those terrible things from IKEA and you end up with a lot of nuts and bolts at the end, even though you think you built something. Those are probably the plasmid genes, and we just don't know where they belong. So we literally, about a year ago, brought into the lab nanopore sequencing, which is long-read sequencing, and it allowed us to identify. Nanopore is complicated and it's expensive, but we said, you know, let's go through our tree, and I don't expect you to see this except for maybe the red lines. We said, let's go through our tree and let's just pick representative different sequence types and let's sequence them by nanopore to get an idea of the plasmid diversity. So just as a reminder, the biggest problem with Illumina versus nanopore, with Illumina, you do get short reads, and when you start assembling, you do have gaps. And sometimes you just can't get overlaps. And the biggest problem, which you should appreciate, any genes or any chromosomal content or plasmid that have large repeats, you can't assemble them. Whereas when you look at nanopore reads, where you're actually on average something between around 15 kb size, you can start literally overlapping fragments or contigs and assembling them. Most importantly, if you have Illumina and nanopore data, then you can not only do the assembly, but the Illumina basically reinforces your assembly because you can actually stitch the short reads Illumina now to your assembled genome. So in combination, when you have Illumina and nanopore or PacBio, you can really start closing genomes and closing plasmids. Okay, this is 39 representative plasmids from our collection, and this is just showing you how complex these are, but I do want you to look at top. So on top is the cartoon of CTXM15, which they all have. And what we learned is that CTXM15 has this little transposable unit in front of them, which also has a promoter. And that's the way we think this thing is hopped around. And that's true about a lot of resistance genes that you'll see on this island. Now, I'm going to show you a little more clear slide next, but I just want you to get a sense of what you're looking at here. We have resistance genes, we have heavy metal resistance, we have other environmental genes on these plasmids, and remarkably, when you look at the resistance, so on top, every red arrow is another resistance gene on this island. We have over 10 resistance genes on most of these plasmids. And as you'll see, and also I want to point out the plasmids on top. So the first 1, 2, 3, 4 plasmids have what are in green here. Those are conjugate, that's a trioperon. So these are conjugated plasmids, whereas there are within our collection non-conjugated plasmids, which are on the bottom, which are also drug resistant. But as you should appreciate, conjugated plasmids, especially with hybrid replicons, really are dangerous because not only can they move from strain to strain, because they have 2 different replicons, they can overcome plasmid incompatibility. If there's a resident plasmid with the same replicon, they use the other one. So these plasmids have really set themselves up for being able to move and spread, and that's exactly what they've done. So this gives you a little better and a little clearer idea of how much resistance these plasmids have. So not only do they have CTXM, they also have other beta-lactamases. They have OXA and TEM, which don't give you ESBL, but contribute to resistance. They have 3 different fluoroquinolone mechanisms. They have a Bactrim, 2 different Bactrim mechanisms, aminoglycoside resistance, sulfonamides. So literally, this is chock full of resistance. And as I'll show you in the next slide, almost every plasmid, these INCA plasmids, have this array. So it's a remarkable drug resistance. And I gave this talk to clinicians recently, and what you try to impress, especially about ID docs, and you'll see this at the end, if you have one single event, meaning the plasmid moves from one strain to the other, all this resistance comes with it. It's a package. So even though you think you're moving quinolone resistance, you are, but everything else comes with it. And that's what multi-drug resistance means. And you'll see that when we talk about literally curing plasmids and bringing back genes to show you this. Now, what I really want you to remember about this talk is not the resistance. You always hear about resistance, you hear about plasmids and transfer. The other thing these plasmids have is environmental factors that allow them to survive. And we'll talk about this at the end. But I just wanted to appreciate that they have heavy metal resistance. They're resistant to silver, copper, arsenic, and tellurium. They also have heat resistance genes, which is called CLPK. They have an iron uptake system. They have various lactose operon. And these are only the things we know how to name. These plasmids have 200 genes. We only know about 30 of them. So clearly, whatever these plasmids are contributing is way beyond our knowledge. But obviously, as you can see, these factors clearly are survival factors. These allow these organisms to survive in the environment. And I'm going to try to impress upon you at the end that the medical or the clinical consequence is not what these bugs want to do. These bugs need to survive, and they survive in the environment. Remember, enteric bugs in your gut end up as sewage. Sewage ends up in water. Sewage ends up in agricultural settings, but it starts in your gut. But the bug really wants to survive in the environment. So it is chock full of environmental factors that really just help it survive in very, very bad situations. Okay. This again, I don't expect you to see what this is just showing you that all the resistance I told you about, all the other environmental factors, they're represented really in the high 90s for every plasmid that's on this list. So almost every strain in our collection, which is about 90%, which have these plasmids, literally almost have all this resistance with it, which is quite remarkable and quite frightening. And remember, these are community isolates causing urinary tract infections. Right? These are not, these are not in the ICU, although I think they are, but the idea is that we have to treat these patients in the community with these very drug-resistant organisms. Okay, so what I've told you in the last 20 minutes is really a snapshot of our collection that we're working with. And again, it's a 1-year collection. We have over 2,000 strains, but it really is just a snapshot. There's 90% of strains we didn't even look at, although given the similarity of everything we have looked at, our expectations is we're just going to see more of what we have. Again, what's unique about this study is it is the community. Obviously, we want to now look at what's in hospital settings as well as long-term care. but we expect it's going to be similar. ESBL is really CTXM15 driven, which is somewhat surprising how much it is CTXM15, and this is also true in isolates that other people reported from Europe. INCF plasmid is the predominant ones, and they're literally in almost every strain. As I said, they harbor multi-drug resistance, environmental factors, they're diverse. And the one thing you need to appreciate is that when you have plasmid spreading from strain to strain, and you have strains spreading clonally, you have two things going on simultaneously, both not good. So we have literally plasmid spreading and strain spreading at the same time. So you have a large burden of resistance. Okay, so the last couple slides, I really just want to give you a little more basic biology of what we can do and try to understand these factors. So just because we identify a plasmid and we see that it has all these genes, we make the assumption that the genes we can name are the cause and effect of resistance or these environmental factors. But we do have the ability now to actually test this in the lab. So one of the things we developed recently was a paper that we published last year in trying to cure strains of their plasmids. Now, historically, people have done curing experiments over the last 30, 40 years, trying to kick out plasmids from a strain. And historically, the way curing was done was either by spontaneous curing, where you just played out a lot of strains and hope for the spontaneous loss of a plasmid, which doesn't happen that frequently, or you treat the strain with heat or some chemical, and you try to kick the plasmid out that way. The problem with trying to do that with any type of way to perturb the plasmid, you usually also perturb the chromosome, and you end up with mutations. So, and years ago when we used to do this, we couldn't sequence the strain. So curing strains years ago was really not it was really an imprecise science and probably caused more damage than we thought of. So we developed a system using CRISPR where we use CRISPR to actually target the replication gene of the plasmid. And by doing that, we can surgically remove each of the plasmids in a strain one by one and ultimately create a plasmidless strain. And what's great about that is once you have the plasmids removed, it's a lot easier to study the chromosome. Plus, you can add back now plasmids to those strains and ask, what does that contribute? So that's what we've been doing with this collection. And so just as an example, we took one of our strains, one of our ST307s, which has this enormous plasmid, and we cured it of that plasmid. And then we looked at the difference. So what you're looking at now is the parent strain on the left, which has the plasmid, and the strain without the plasmid. And we went back and did antibiograms, and not surprisingly, what we expected was that we would see an incredible reduction in resistance, which is exactly what we saw. So as you can see from the beta-lactams, so these are second, third generation, fourth generation. Ultimately, the MIC goes from being resistant to going to being susceptible. So when you remove this plasmid from the strain, it almost becomes a lab strain. It's incredibly susceptible, and essentially now you can use it and manipulate it much like you could manipulate a lab strain. I also want you to focus at the bottom, and I'm going to show you two more slides about it, but things like arsenic resistant, not surprisingly, changed dramatically. Heat resistance we showed was also on the plasmid, as well as the iron uptake system. So the plasmids, what we thought clearly does or do contribute to resistance. Now, what we've also been able to do, and I don't have a slide of it, we've also now been able to go back using CRISPR and not only remove the plasmid, now we've been able to just knock out individual genes on that plasmid and then ask how much does that single gene contribute to resistance. So when we knock out CTXM15, which we have, and put that plasmid in, it has everything else but CTXM15, and what goes away is not surprising, is the second and third generation resistance, whereas they're still resistant, obviously, to cefepime and the fifth generation. So we can literally now do gene knockouts and really show cause and effect for almost every gene we know. And we've done it now with almost, well, we've done it with the heat resistance genes, we've done it with the iron genes, and we've done it with some of the heavy metals. So here's an example of the wild type versus the cured, and this is just doing heat experiments. So obviously at 37 degrees, they both look fine, but as you go up to 45 or 53 degrees, the wild type, which has the plasmid and has the CLPK gene, it could withstand a lot of heat. And if you just think about it again, it doesn't need, when these bugs have these plasmids, you don't need the whole plasmid or the whole strain population doesn't have to survive. When you have one of these heat resistance genes, the population is probably being killed, but you do have survivors. And that's allowing these bugs, again, to survive in really bad environments, including things like heat, or in this case, low iron, where it's the same exact experiment, but now we're using low iron media. And as you can see, when you don't have the plasmid, the bottom one where the arrow, they don't survive as well. So again, the survival mechanism is really what I think is the important part of these plasmids, because it's ultimately allowing these bugs to persist and then do what they do, either clinically or in various settings. Now, I'm going to leave you with this slide. So this really surprised us and actually struck me more than anyone else in the lab, because whenever we think about resistance, as I said, when you look at these plasmids, you look at these strains, we think clinically. We think about the medical problem. Well, this paper came out of Norway this year in 2024. And this study, they looked at a sewage plant in Norway. And they isolated Klebsiella out of the sewage plant. This Klebsiella is an ST307. Sounds familiar. This plasmid that came from this ST307 in the sewage plant is identical to the plasmid I just showed you. It's about 10 kb difference. It has every gene that plasmid we took from a urinary tract infection has. So this just really reinforced the idea that the factors I told you, the factors like arsenic resistance, heavy metal resistance, and heat resistance, and all these other factors are allowing these bugs to survive in these types of environments. And therefore, as they persist throughout nature and in these other settings, they're just going to be more prevalent. And then they're going to be ultimately the opportune bug that is going to cause a disease such as a urinary tract infection in the community. And what I think the resistance is doing is not really to deal with the medical side, but the one thing you need to appreciate, and I literally looked it up last night, something like fluoroquinolones, like ciprofloxacin, which we use, tens of millions of dollars, if not $1 billion drug. Ciprofloxacin does not break down in the environment. We have ciprofloxacin in our water system. We have ciprofloxacin in those sewage plants. So even the resistance mechanisms, which we think are really just for the clinical setting, no, the resistance mechanism on these plasmids actually help them to survive also in the environment, because the environment, unfortunately, has a lot of antibiotics that we spill over from either the medical side or just flushing them down the toilet. And therefore, even resistance is probably more to do with their environmental survival than what they do in terms of, causing havoc for doctors trying to treat them. So I think we've been thinking about resistance maybe backwards. It's really the bystander effect that resistance is a problem, but that's not what these bugs want to do. These bugs want to survive, and they survive not in your gut or in a urinary tract infections, they survive in the environment. So with that, I don't do any of this work. My job is really to make the deals with Quest and to try to get the lab to do these. But I must say, without Quest, which are my colleagues on the left, these studies cannot be done. We've recognized, so literally as a result of this study, we just worked out a program with Quest where we're going to look at strep pneumonia isolates, same exact study, but instead of Klebsiella, we're going to ask them to send us invasive strep pneumonia isolates from both children and adults. And we're going to do a serotype study to understand the vaccines that are being used. And again, no one's done these studies. They haven't had a surveillance on strep pneumonia across the US since 2017. And the reason is it's just difficult to collect isolates from the community. And when you have someone like, or some organization like Quest or any large diagnostic lab, it really becomes a gold mine to mine. And I must thank them 100% because these studies cannot be done without them. These are the folks in my lab who have done all this work, which I really think we're going to try to get this paper out sooner than later to make it timely. But literally, we now have a good baseline to work from. And that 10% we've collected is really just a snapshot. Now we can use that as a baseline with objective sequence data to really see what's going on in hospitals, long-term care facilities, and hopefully other people who do their studies can use our data set as a template to compare it to. So with that, I'd be happy to answer any questions.
Switch it on.
Hey, Barry, that was really fun.
Thanks.
So the question for you is, do you think these plasmids are common in non-pathogen bacteria? And so, you think that maybe there's just response genes help perhaps like beneficial bacteria persists, and you know, in the case where you are curing these plasmids, perhaps you can have an adverse effect on the health, for example, by depleting beneficial bacteria.
I'm a little hard here. I didn't hear the first part of the question.
Are these plasmids common in non-pathogenic bacteria, perhaps even beneficial bacteria?
Yeah.
Right.
Yeah, I think that's a good question. If I had to guess, my answer would be yes, but we don't really know. I mean, it's funny, the more we start to see this, I don't even know, and we're going to do the study, hopefully. You know, one of the questions that we've been asked is E. coli, which is a much greater pathogen than Klebsiella. You know, Klebsiella is a pain in the neck. It only became, quote unquote, a major pathogen as it became really resistant. But E. coli is a bloodstream organism which really can kill you very easily. I don't even know, and we're going to do the study, whether these INCF plasmids now are predominating in E. coli. No one's even looked at ESBL resistant E. coli as much as we should have. So getting back to your, I have a feeling that if we look at non-resistant strains, you wonder if the backbone of some of these plasmids without the resistance genes are there. And it's a good question. And we literally looked the other day at some of the rarer plasmids in our collection, and a lot of them don't have the entire spectrum of this resistance, what we call the resistance region, but they do have all the other environmental ones. So I think we need to look at, I mean, it's interesting when being a staff biologist, everyone sequences MRSA, everyone studies MRSA, but MSSA is actually much more interesting and much more diverse and will kill you just like MRSA will kill you. But a lot of our molecular epi has been driven by the clinical side and resistance. So we do have a real, I mean, if you look at the genomes in the public domain, they're mostly drug resistant. We do have a bad bias, and it's because that's the way you get funding, et cetera, et cetera. But I agree with you. I think we need to start looking at sort of the non-resistant strains and look at other non-clinical strains to see where the reservoir is. Because I have a feeling these INCF plasmids are probably out there in lots of different organisms that we don't think about as clinical problems. Yeah.
Hi, great talk, thank you. I have a question about, so plasmids are of course very efficient at spreading these resistant genes and these effects of all these other genes. Have you looked at any other like genomic mutational like hotspots or certain other changes in the genome itself that could also facilitate the resistance mechanism?
So are you talking about the actual resistance spectrum or acquiring resistance?
The resistance strains, the isolates have you looked at the genomic background, the genomic composition of that, and were there any variations there from?
Okay, yeah.
the non-
So quinolone resistance as an example, right? So the plasmids have QNR and this other AAC gene. But quinolone resistance is primarily mutations in gyrase and PAR-C, which are chromosomal housekeeping genes. So you're absolutely right. So not only do these plasmids come into strains that are already resistant due to chromosomal mutation, there are probably, we've just talked about this literally the other day in the lab. So things we're probably missing are things like pumps and other regulators. So yeah, so one of the confounders, which I didn't talk about from time, cefepime resistance. 50% of our isolates are cefepime resistant. We think we know what cefepime resistance is based on these beta-lactams, and yet the ones that are resistant, the ones that are susceptible, look alike. So I think, to get to your question, I think there's factors in the chromosome, or I think there's crosstalk between the plasmid and the chromosome that we don't appreciate that is probably contributing to some of the resistance we can't explain. And that's what becomes nice about these collections. And by taking away the plasmids, it just allows us to focus a little better, because otherwise by having plasmids in strains, it's such a confounder trying to figure out what's causing what. So I agree with you. There's probably many more things in the chromosome and or plasmid genes we don't know about or name. You know, the funny thing about genomics is we study things we know the names for, right? But obviously, just because we don't name the gene doesn't mean it's not important. It's just a stupid mindset we have that we always focus on things, because no one's going to fund things when you say, I'm going to look at things we don't know about. But that's what we should be doing. So, but it's a good question. Thank you. Yes. Sure.
Your point on the environmental residues and how much that might be driving this. Now, partly we can find each other these because of our wonderful sensitivity of organic communication. Are those concentrations in the wastewater treatment plant or the Raritan River high enough to actually drive resistance of strains?
It's a great question. So it's funny. I looked at a review article on quinolones last night, and it talked about the concerns of quinolones in the environment to do with mutagenesis of, fish and crustaceans and everything else. It never got to bacteria. But I agree with you 100%, and it could be not only about the levels, but it also could be that you have 6 or 7 different antibiotics in the water that collectively, they're at the subclinical level. But as you know, and I think everyone here, that's the problem of building more resistance. If you don't actually have enough bugs to kill them, they're just literally surviving, and they're being teased with enough antibiotic, to maybe even cause more mutations. People have been doing, as we know from COVID, people have been doing a lot of water testing. I just wonder what they should go back. There's so many things. I would love to understand the arsenic resistance. I've been talking to folks who actually come from friends from Montana and some from Western Pennsylvania, where they do a lot of mining. And apparently arsenic leaches all throughout when we do mining. So again, how much arsenic resistance do these bugs actually provide in terms of survival? And it seems pretty scary when you think of these sewage plants, what goes on in a sewage plant. So I've been trying to talk to it's hard to do environmental studies, I've learned. You get involved with the EPA, you get involved with all these companies and or public health departments. It's tricky to ask, can we just sample your water to look for this? But I think it needs to be done. And this, the more we see just what you were saying about what is the level, it shouldn't be that hard to test, right? And maybe that's what we need to start thinking about. We need to look at it from the other side, not from the clinical side. So thank you. It's a good question. Okay.